This is the Alzheimer's disease SNP-CHIP test presentation by Group 8 with Aaron Kim, Christy Kim, Aidan Rathbun, and Sean Collins. The name Alzheimer's disease was coined by Dr. Alois Alzheimer, the original discoverer of the disease. According to a paper published by UCLA on the history of Alzheimer's, Dr. Alois Alzheimer described how he found a patient named August D. to have a constantly worsening memory, and he observed that, upon her death and autopsy, her brain had significantly decayed. Alzheimer's is the most common cause of dementia, a general term for memory loss and other cognitive abilities serious enough to interfere with daily life. Alzheimer's disease accounts for 60 to 80% of dementia cases. Because it is a degenerative disease, it is more common in elderly populations, being present in 1 in 10 people who are 65 and older, though it is important to note that Alzheimer's is not a natural part of aging. Currently, there is no cure for Alzheimer's disease, though there are treatments available. It is estimated that around 70% of the disease's development is attributed to genetic causes, and 30% is attributed to environmental causes. The proteins that are altered in people with Alzheimer's are primarily APO, PSEN1, and PSEN2.
The 23andMe test for Alzheimer's disease searches for the E4 variant in the APO gene associated with late-onset Alzheimer's disease. Yes, the E4 variant of the APO gene significantly impacts the structure and function of the apolipoprotein E, which is associated with an increased risk in acquiring Alzheimer's. This protein helps control the levels of cholesterol and fats in the blood. The apolipoprotein is primarily in liver cells but can also be seen in the central nervous system and brain cells. Having at least one ApoE4 gene doubles or triples the risk of getting Alzheimer's disease. Some people have 2 ApoE4 genes, one from each parent. Having 2 genes increases the risk of getting Alzheimer's disease even more, about 8 to 12-fold. Yes, there are other variations in other genes that impact the risk for acquiring Alzheimer's. Those genes include PSEN1, PSEN2, and APP.
Moving on, the 23andMe test for Alzheimer's only checks one variant in the APOE gene as seen on the 23andMe website. However, one benefit of using our test would be that we would check for additional genes that could help evaluate a predisposition towards Alzheimer's. The variations of the APP, PLD3, TM2D3, and TREM2 genes would be included in our test, therefore being more accurate than the 23andMe test. To give an example, the variation of the TREM2 gene, RS759326228, is linked to significantly increasing the risk of late-onset Alzheimer's. This alters the protein structure because it substitutes the amino acid histidine to arginine at position 41161514. According to the National Institutes of Health, the typical function of the TREM2 protein regulates purinergic receptor-mediated calcium signaling and motility in human iPSC-derived microglia. That may sound complicated, but in other words, the TREM2 gene helps to control microglial cells in the central nervous system, which affects the calcium levels and cell movement. In terms of determining risk, the odds ratio was 2.9 in one study, therefore meaning that the carriers of the variant had 2.9 times higher odds of developing Alzheimer's compared to non-carriers. Nature Reviews Neurology also claims how the rare loss of function variants in the gene encoding triggering receptor expressing on myeloid cells to increasing the risk of developing AD or Alzheimer's in humans. So, why is this important? It shows how checking for more variations in genes beyond just the APOE gene will give people more security and safety, knowing that they are fully aware of the risk for the disease.
The test for Alzheimer's disease has many considerations because of the arguments against the widespread testing. For example, many factors of considerations for the genetic test may come up like ethical and practical scenarios. Because of these, there might be some reasons on why someone would not take the genetic test. Limitations of known alleles impacting the trait of Alzheimer's are mostly the ApoE4 allele, because this is one of the most common genetic risk factors for the disease. If a patient has a copy or 2 of this allele, it can increase the risk of developing Alzheimer's. However, having this allele does not directly mean the patient will develop the disease, which is quite unfortunate as some patients are falsely diagnosed with Alzheimer's. The predictive value of the ApoE4 allele results in a possible risk of Alzheimer's, but it does not confirm the development of the disease. This is why some positive test results may not be accurate because of how Alzheimer's disease never fully develops. Other unknown variations like the environment and genetics can also have an impact on the progress of Alzheimer's.
Lastly, there are also some ethical implications for testing positive for a genetic disease. People will be more stressed and unable to enjoy life as much knowing about how horrible Alzheimer's can be. It may cause tension and frustration between families who now live in fear of Alzheimer's if their relative tested positive. It can also be expensive to maintain the disease, even if there is no cure. Furthermore, the consumer may also want to consider the misuse of genetic misinformation by businesses and insurance companies who can exploit and charge higher premiums.